October 1944, at Glennon General Hospital in Okmulgee, Oklahoma, a U.S. military medical facility dedicated to treating German prisoners of war. A young German soldier lay on the operating table with a severe wound to his thigh. Having fought desperately on the European front before being captured only days earlier, the bullet having torn through muscle and left the wound badly infected, oozing pus and reeking of decay, and in his mind, vivid memories surfaced of comrades back home, whose arms or legs had been amputated because of infection. So he shut his eyes tightly, almost certain he was about to lose this leg if not his life itself. And even in his half-conscious state he could still make out footsteps and English voices as U.S. Army medics prepared for what he believed would be a last, desperate operation. Yet what stunned him was that the Americans did not rush to amputate, because instead he caught a glimpse of a doctor in a white coat taking out a small vial of pale yellow powder, carefully sprinkling it onto the wound after cutting away the dead tissue, then injecting him with a dose of medicine before stitching the wound closed, and he could hardly believe his eyes. The enemy was trying to save his leg, unaware that he had just been treated with a new American miracle drug, a miracle that would not only save his leg, but would quietly go on to save the lives of hundreds of thousands of soldiers in the most devastating war of the century. The German soldiers' astonishment did not come from the enemy's compassion, but from a far harsher reality. Throughout most of modern warfare, wound infection had been the most efficient killer of all. Long before World War II, infection was a death sentence for generations of soldiers. In World War I, more troops died from infection than from bullets or artillery. For a long time, doctors could do little more than wash wounds with harsh antiseptics, cut away infected flesh, or amputate limbs outright to stop gangrene from spreading. A turning point came in 1935, when German pathologist Gerhard Domach discovered sulfa drugs, the first synthetic antibacterial agents. Domach tested the new drug on his own daughter after she developed a severe infection from a needle wound, and she recovered without the need for amputation. Sulfa was quickly hailed as a miracle of its time, capable of saving lives from terrifying infections such as pneumonia and erysipelas. By the early years of World War II, the idea of treating infection on the battlefield was still more preventive than curative. Every American soldier carried a packet of sulfa powder in his individual first aid kit, the most advanced synthetic antibacterial medicine Western medicine possessed in the late 1932. When wounded, soldiers were instructed to sprinkle the powder directly into bullet or shrapnel wounds, hoping to slow bacterial growth before reaching a medical station. The sight of soldiers bending down to pour sulfa powder into their wounds became a familiar image in early wartime documentaries, reflecting the belief at the time that humanity had finally found a way to control infection on the battlefield. Reality, however, quickly proved that sulfa was no miracle cure. Its widespread, frequent, and sometimes excessive use allowed many strains of bacteria to adapt and develop resistance. On the battlefields of Europe and North Africa, military doctors soon realized that sulfa powder could only delay infection in mild cases, while in deep, crushed, or heavily infected wounds, its effect was often negligible. Many wounded soldiers, despite having sulfa applied from the very beginning, still fell into cases of gas, gangrene, or blood infection. Faced with such situations, the familiar choice did not change. Amputate the limb to save a life, or watch the patient slowly die in agony. It was precisely in this context that military medicine began to understand that modern warfare required something stronger, a solution that did not merely delay death, but could truly destroy bacteria. The drug humanity was searching for had in fact appeared before the war, but under entirely accidental circumstances. In 1928, British scientist Alexander Fleming discovered that a mold of the Penicillium family released a substance capable of killing bacteria in his laboratory. He named the substance penicillin. For many years, however, the discovery existed only as an intriguing scientific note. Penicillin was extremely difficult to purify, hard to preserve, and nearly impossible to produce in quantities large enough for widespread use. Within the medical context of the time, it was seen as a brief flash of promise. Interesting, but not practical enough to change anything. Everything truly changed only when two scientists at the University of Oxford, Howard Florey and Ernst Chain, decided to return to penicillin research in 1940. They did not view it as a curious accident, but as a potential medical weapon. Early experiments showed that penicillin could save patients suffering from severe infections against which sulfa was completely powerless. Wartime Britain, however, lacked the industrial capacity to produce penicillin on a large scale. German bombing, shortages of raw materials, and a lack of manpower quickly pushed every effort to its limits. The true turning point came when the United States entered the picture. In 1941, Florey brought samples of the penicillin mold to the United States, seeking support from the world's largest pharmaceutical industry. The American response was almost immediate. Laboratories, drug companies, and military agencies plunged into an unprecedented race, transforming penicillin from a laboratory curiosity into a medicine that could be mass-produced. American scientists quickly refined cultivation methods, moving from crude culture trays to deep tank fermentation techniques, multiplying output many times over in a short period. Penicillin, once a rare substance, gradually became an integral part of the wartime logistics system. By September 1943, U.S. penicillin production was sufficient to meet most of the military's needs across multiple fronts. By June 1944, ahead of the Normandy landings, the Allied forces had stockpiled enough penicillin for the entire Operation Overlord. For the first time in history, military commanders entered a major campaign with the assurance that thousands of wounded soldiers could be saved after being hit. Penicillin was sent to the front lines in the form of injections and topical treatments, becoming an indispensable component of field hospitals and hospital ships. The soldiers who landed on the beaches of Normandy carried not only weapons and ammunition, but also carefully safeguarded vials of penicillin. They understood that if they were wounded but survived the initial moment, their chances of returning home were far higher than those of their father's generation in World War I. For military doctors, the effectiveness of penicillin was nothing short of revolutionary. Wounds that once had to be left open or treated by amputating limbs could now be safely stitched closed. Infections were controlled rather than allowed to spread. Wounded men recovered faster, hospital stays grew shorter, and rates of gangrene fell to levels never seen before. Penicillin did more than save individual lives. It fundamentally changed how armies confronted the consequences of war. Meanwhile, on the other side of the front lines, the German military largely stood outside this medical revolution. Despite possessing one of Europe's most advanced scientific traditions before the war, Nazi Germany's research and industrial systems reacted slowly to the rise of biological antibiotics. Confidence in domestically produced sulfa drugs, combined with the strain of a prolonged war and relentless Allied bombing, severely delayed efforts to produce penicillin. It was not until October 1944 that Germany managed to produce its first batches of penicillin, by which point the country's infrastructure had been so heavily damaged that large-scale deployment was no longer possible. The consequences of this delay were starkly visible on the battlefield. For most of the war, German soldiers had little more than sulfa to treat infections. At first, the drug showed some effectiveness, but as the war dragged on, cases of bacterial resistance increased steadily. Many wounded men died not from their bullet wounds, but from infections that could not be brought under control. The case of Reinhard Heydrich, who died in 1942 from infection following an assassination attempt, is often cited as a telling example of this shortfall. In 1944, Adolf Hitler himself narrowly avoided a similar fate after the July 20 bombing. He survived only because his personal physician managed to obtain an extremely rare supply of penicillin, while many other officers wounded in the same attack did not. The paradox was unmistakable. To save its leader, Nazi Germany was forced to rely on an invention developed by its enemy, while millions of ordinary soldiers never had any chance of accessing the drug. Without penicillin, German doctors continued to depend on crude measures, resulting in rates of gangrene and death far higher than those seen among the Allied forces. That disparity created a profound gap between the two sides, not only in weapons or tactics, but in science, industry, and the ability to preserve human life. Penicillin did not decide the outcome of any single battle, but it decided who could survive after the battle was over. And in a war of attrition like World War II, that advantage carried strategic weight equal to that of any weapon. All of those systemic differences from laboratories and production lines to the battlefield itself now converged in one very specific moment, on the body of a German prisoner of war, lying motionless on an operating table inside a U.S. prison camp. The surgery had ended without him knowing when. As the anesthesia wore off, he slowly opened his eyes and found himself in the recovery ward, his leg still intact and carefully bandaged. The fever caused by infection had subsided, and the wound no longer appeared swollen and inflamed as before. A U.S. Army doctor, sweat still beading on his forehead after the operation, stepped closer, gently patted his shoulder, and offered a reassuring smile. Even though the language barrier kept him from understanding every word, he realized one thing clearly: his life had been saved. He had not been left to die, nor treated brutally as he had feared. The enemy had saved him using the most advanced medical science of the time. Lying in the hospital bed, the German soldier was overwhelmed by emotions he could barely put into words. Relief at having survived, gratitude that his leg was still there, and at the same time confusion and disbelief. He could not stop wondering, why were the Americans going to such lengths to save me? What kind of medicine could do something like this? Around him, other German prisoners were being treated as well. Some patients were brought into the infirmary in conditions that, on the German side of the battlefield, would almost certainly have been considered beyond saving. Severe pneumonia tightened their chests, their breathing became short and rapid, and their lips turned blue from lack of oxygen. In the memories of many, pneumonia under wartime conditions meant little more than waiting, because sulfa could offer only limited support, while lung infections often progressed faster than any effective intervention. Here, however, those patients were given penicillin on a regular schedule, not a single dose and no more, but according to precise amounts and timing. After only a few days, their breathing began to steady. The fever gradually subsided. Men who had been delirious from oxygen deprivation started to regain clarity, able to speak, able to sit up in their hospital beds. For those who had watched comrades slowly die of pneumonia during the European winters, this change was almost impossible to believe. In other beds, shrapnel remained the greatest threat. Many had been wounded by small fragments that lodged deep in the body, carrying dirt, fabric, and bacteria into muscle tissue. Under conditions of medical scarcity, such wounds often turned into prolonged sources of infection. In the American prison camp, surgery was carried out thoroughly. Shrapnel was removed. Dead tissue was carefully cut away. Wounds were stitched closed instead of being left open. And afterward came continuous doses of antibiotics to prevent bacteria from flaring up again. What astonished many was not the operation itself, but what followed. Wounds that had been accustomed to swelling and oozing pus began to dry. The familiar stench of decay was absent. There were no lingering fevers. Recovery unfolded in a way most of them had never witnessed before. In the hospital wards, small conversations began to emerge. At first they were scattered questions, half belief and half doubt. What was this drug? Why was it always available? Why was it injected again and again, always on time? Some whispered that perhaps it was only a temporary measure, that their condition would worsen again after a few days. Others suspected it was a form of medical experimentation, because the past had taught them that the defeated were rarely treated kindly. These doubts were not unreasonable. War had conditioned people to expect the worst. But war also taught another lesson. The body does not lie. As day after day passed, and those lying in hospital beds did not deteriorate, but instead improved, skepticism began to waver. Bandages were removed without the overpowering stench of pus. Wounds began to form healthy new skin. Fevers disappeared. Men who had believed they would never stand again began to sit up. Then practice taking their first steps. When this pattern repeated itself often enough, it could no longer be dismissed as individual luck. It became a shared reality. And once that shared reality took shape, doubt naturally faded, giving way to something deeper: astonishment. That feeling spread quickly throughout the infirmary. No announcements were needed. One only had to look around. Here, the severely wounded were not abandoned. Here, infection was no longer a death sentence, hanging over every injury. And here, the medicine being injected into their bodies seemed to come from another world altogether, one in which medicine had crossed limits they had once believed impossible. This sense of astonishment became even more pronounced when German military doctors who had been taken prisoner entered that environment. Under the Geneva Conventions, they were permitted to help care for their fellow soldiers under the supervision of U.S. Army medical staff. At first, many of them approached the situation defensively. They were accustomed to working under conditions of scarcity, where every decision involved painful trade-offs. Yet it took only a short time for the differences to become too obvious to ignore. The wards were clean. Surgical instruments were complete. Equipment functioned reliably. And above all, medicines were not something that had to be begged for one dose at a time. Dr. Friedrich Bauer, a former German Army medical officer and once the chief surgeon of a German division, later recalled that he was nearly stunned when he opened the medicine cabinet in the prison hospital. Inside was penicillin, not as a rare substance locked away for exceptional cases, but as an ordinary part of daily treatment. He witnessed this antibiotic being administered even for infections that were not yet life-threatening, something that would have been unthinkable in the German medical system at the time. Back home, doctors had to ration every Sulfa tablet, every bandage. Here, the Americans were using the most effective medicine they had, even on their enemies. From a professional standpoint, what shocked the German doctors was not merely the abundance of supplies, but the approach itself. Penicillin allowed surgeons to make far less destructive decisions. They could close wounds instead of leaving them open. They could preserve limbs rather than amputate them. To be safe, they could trust that post-operative infections would be brought under control. For those who had long worked in desperation, where every operation was a gamble, this shift was nothing short of revolutionary. Those observations quickly spread from the medical staff to the men lying in the hospital beds. When doctors from their own ranks acknowledged that this drug truly worked, confidence within the infirmary became more tangible. Whispers about experimentation faded away. In their place came stories of recovery, of limbs saved, of the possibility of surviving without carrying lifelong disability. In the context of war, such realities carried far more weight than any promise ever could. The humanity and attentiveness they experienced stood in stark contrast to the image of the enemy that Nazi propaganda had long drilled into them. Not through grand gestures, but through small, repeated actions. Tetanus shots given even for wounds that seemed insignificant. Bandages changed on schedule. Temperatures and signs of infection monitored closely. It was this consistency that proved most astonishing. It showed that what they were witnessing was not momentary compassion, but a system operating according to principle. For many, the moment of realizing this marked a profound psychological turning point. They had not only been saved, but were forced to confront an uncomfortable truth. The enemy was not only superior in weapons, but also in the ability to save lives. Penicillin became the clearest symbol of that difference, not because it was magical, but because it was used correctly, in proper doses, and within a system strong enough to sustain its effectiveness. And it was inside the quiet hospital wards of an American prison camp that this truth revealed itself most starkly, leaving many unable to believe what they were seeing. The effectiveness of the drug used by U.S. Army medical staff required no grand declarations. It revealed itself most clearly in the first hours after surgery, when the bodies of those just taken off the operating table began to respond in ways most of them had never witnessed throughout the war. In the recovery wards of American prison camps, many German prisoners of war awoke from anesthesia with the same sense of disbelief. The pain was still there, but the thing they feared most had not happened. Their arms and legs were still intact. Wounds that had been severely infected did not continue to spread. High fevers gradually subsided, instead of rising as usual. For those accustomed to watching gangrene advance hour by hour on the battlefields of Europe, this change lay almost beyond the logic they had come to accept. In the mindset of men who had once worn the Wehrmacht uniform, a deep infected wound had almost always marked the beginning of a series of desperate decisions. Amputate the damaged limb, or accept death from blood infection. These memories came not from textbooks, but from their own experiences and those of their comrades. They had seen too many arms and legs sawn off in under-equipped field hospitals. Yet here, in the prison camps of the enemy, surgery followed an entirely different logic. U.S. Army surgeons carefully cut away dead tissue, closed wounds with stitches, and then monitored their patients with regular injections afterward. There was no frantic rush, driven by fear of imminent death. No moment on the operating table where survival had to be weighed against the loss of a limb. Infection, once regarded as an irreversible sentence, was now being brought under control. That sense of shock quickly spread beyond the experience of any single individual. In the hospital wards, those taken prisoner exchanged quiet, fragmented conversations, mixing doubt with hope. Some had been admitted with severe pneumonia, their breathing weakening day by day, only to find themselves able to sit up again after a short period of treatment. Others carried shrapnel lodged deep in their bodies, convinced that such wounds would end in prolonged infection. Yet they recovered without the familiar signs of gangrene ever appearing. These individual cases, taken together, formed a broader picture that many could no longer deny. Something was operating differently within the medical system on the other side. Gradually, one name began to surface more frequently in the infirmaries: penicillin. For most of those who had once stood on the opposing side of the front lines, it was not a familiar concept. If they had heard of it at all, it was only through vague, fragmented references, unconnected to any real experience. At first, skepticism remained. Some could not believe that the U.S. Army would willingly use such a valuable drug on defeated enemies. Others even feared they were becoming subjects of medical experimentation. But those doubts were quickly overwhelmed by reality. With each passing day, more wounded men recovered. Bandages were removed without the overpowering stench of pus. Prolonged fevers disappeared. Wounds dried and began to form healthy new skin, not over months, but within days or a few short weeks. This change was especially shocking to the German military doctors who had themselves been taken prisoner. Under the Geneva Conventions, they were allowed to help care for men from their own side, under the supervision of U.S. Army medical staff. That access enabled them to observe the entire treatment process firsthand, from the operating room through post-operative recovery. What they witnessed was not merely an abundance of equipment, but an entirely different approach to infection. Penicillin was not used as a last, desperate measure, but as a standard part of treatment. Dr. Friedrich Bauer, a former German Army medical officer, later described a sense of near disbelief at seeing this antibiotic used routinely, even for infections that were not yet life-threatening. From a medical standpoint, the effectiveness of penicillin revealed itself with cold clarity. It suppressed the growth of infection-causing bacteria, halted the spread of gangrene, and allowed surgeons to close wounds rather than leave them open or amputate limbs. This not only reduced mortality rates but also dramatically shortened recovery times. Men who had been certain they would lose the ability to work for the rest of their lives could now stand, walk, and regain their strength. On a larger scale, this meant that thousands of lives were preserved not through weapons or tactics, but through a single drug that fundamentally altered the nature of warfare medicine.